morning, everybody, and welcome to SynBioBeta Live. This is, believe it or not, our first event of 2021, and how good it is to get 2020 behind us and look forward to the green pastures of the synthetic biology revolution, which are being ushered in as we speak. We have a very exciting event that's happening today with a number of guests who I'm going to introduce shortly. But as people are just arriving and we haven't done an event in a while, I'd love for you to type into the chat box and tell us where you are joining from. And if you have the option to share it with the panelists, then they'll be able to see where you're joining from as well. And this is a live interactive event. And so we'd love for you to ask questions later on. And we've got a good period where we can do that. So type into the chat box where you're coming from and where you are based and what you do. That would be great as well. And also we're going to be taking live Q&A later on. So you can ask us any questions or ask any of the panelists any questions. So if you're just joining us, then welcome to SynBioBeta Live. This is the first event of the year. My name is John Cumbers. I'm the founder and the CEO of SynBioBeta. And today we're going to be doing an event all around the Internet of connected biological things, thinking about how you can automate your research processes and how you can automate it across quite large scales as well. And the sponsor of today's shows is Strateos, and we are joined by Peter Lee, who is the Director of Science and Technology at Strateos. Hi, Peter, how are you?
Doing well, yeah. Struggling with the muting and unmuting, but doing great and excited about the panel today.
It's all good. So before we get started and introduce everybody, can you tell us a little bit about Strateos? What does Strateos do?
Yeah, Strateos works at the intersection between science, automation, and software, and really thinks about how we can use software tooling and software mindset to accelerate scientific discovery. We have a one interface, one cloud-enabled interface that allows scientists from all over the world to submit experiments to us. We process them on robust, reproducible automation and return data as well as other outputs back out to our scientists in the field. So that really allows customers from all over the world to access world-class, robust science.
Excellent, wonderful. I'm going to go around and introduce the rest of the panelists that we have today, but the thing that binds everybody together is an exciting project which was funded by DARPA. And here, who works for DARPA is Susan Buckhout-White. So Susan, maybe you could tell us about this little project, please, or big project.
Great. Yeah, thank you. Yeah, I work for Griffin Technologies, and I help DARPA run the SD2 program, which stands for Synergistic Discovery and Design. And the goal here was to be able to accelerate the speed of design in domains that sort of lacked foundational understanding, SynBio being one of them, using AI methodology and high throughput experimentation. And so we set up the program in a way that distributed the portions of science, so the experimentation, the analysis, the computation, as well as the design into individual components. And it is a highly collaborative program, and in doing so, we are able to determine the areas that need additional technology in order to support easy distributed science across
Excellent. Great, thank you, Susan. So we're going to go around and just do brief introductions of everybody who was involved in the project. Daniel Bryce. Daniel is the Senior Researcher for Smart Information Flow Technologies, or SIFT. Daniel, could you tell us what was your role in the project and what does SIFT do?
Sure. So SIFT is a small business based in Minneapolis. We do a lot of work in AI, and my background is in AI. So what I brought to the SD2 program is research on automated planning and resource allocation, as well as machine learning. And so what we've done is we've sat kind of closest to the lab with Strateos, where we are doing work on taking large experimental requests and boil those down into more manageable bytes that can be run as runs at the laboratory. And how do you go about doing that resource allocation in order to accomplish that? And then on the other side, we have contributed work on analyzing the data coming out of the lab and producing data products from that. So that's kind of in a nutshell what we've been doing in SD2.
Excellent. And so, Susan, you just described the reason for this SD2 project, which is that there's a lot of disparate information and research projects which are quite involved and spread across multiple people, multiple sites. What you're trying to do is really kind of trying to apply an engineering discipline overlay on top of that, so that, and particularly around biology, it's a very complex system, a lot of siloed knowledge. So you're trying to create this abstraction layer where different parts of the research process can interface in interoperability. They can have interoperability standards, they can pass data, they can pass instructions. Did I kind of summarize it correctly?
Exactly. The concept of being able to engineer in a way, and instead of basing it on models and theories, what we're hoping is that through sort of data-driven methods and use of machine learning, AI, and advanced analytics, that those coupled with the high-throughput automation will allow the engineering approach to areas like synthetic biology.
Great. And you mentioned that DARPA has funded a number of these areas where the problem exists. It's not just in biology, but clearly biology, surely we're at the top of the pile for horrible complexity and difficult to engineer. But I'm curious if that's true, but I'm also curious, who else did DARPA fund? What other kind of industries have this problem?
So biology is the core of what we're working on. It's the hardest problem. We also have a group working on material science and materials design, particularly in solar cell chemistry. And then there have been some other efforts and side efforts using our methods applying to other things in areas as diverse as neuroscience and other, foundational efforts in those types of fields.
Great. Steve Hasse. Steve is a professor at Duke University, a professor of biology. And Steve, your role in the project, you were one of the people who were coming up with some of the pathways that were being built. Is that right? Did I characterize you correctly?
Yeah, so the Duke group largely is made up of folks from a number of different universities, and our expertise is in mostly systems biology. So where our aim is to understand evolved circuits. And so what we did in the program was try to bring some of the tooling we had, some of the expertise in understanding evolved circuits to the process of engineering circuits that would be robust across growth conditions.
Great, excellent. And we'll come back to how some of the projects were chosen and what some of the specific projects shortly. Mark Weston. Mark is the co-founder and CEO of Netrius. Could you introduce Netrius to us and tell us where you play in this wonderful stack of bioengineering that we're describing?
Yeah, sure thing. Thanks, John. So Netrius is a small business. Internally, I direct the development of a platform to transform heterogeneous life sciences data into AI-ready data frames. And so on SD2, I support the program in the role of a system integrator. And so all this discussion around the complexity and engineering and metadata and harmonization, I focus on developing lab-agnostic technology to both harmonize metadata and to do multi-omics data integration for the program. So taking data from across the labs, whether it's Peter at Strateos or Diveena at Ginkgo or data from Steve at Duke, translating all of the terms that we see in those data sets, getting harmonized data frames into the hands of analysts on the program so they can understand what happened in an experiment and figure out, the next turn of the crank, what we want to do next.
Great, excellent. And last but not least, Diveena, you're at Ginkgo Bioworks, and Ginkgo is a big part of this project as well. Help us understand the role that Ginkgo was playing.
Yes, hi everyone. Ginkgo serves in the capacity of a foundry provider. We enable performers in this program to leverage some of Ginkgo's high throughput pipelines, be it to do strain construction or use our next generation sequencing platform to do lots of DNA and RNA sequencing type work. And so we work with the performers on the program to help them design experiments that would run efficiently and use our capabilities, and then provide those data to the performers and let them utilize it for machine learning models and other exciting work they do within the program.
Great, excellent. And just as a reminder to everybody, this is a live event and we do welcome live audience Q&A. If you've got questions for the panelists, then please raise your hand and we will bring you on to ask it yourself. And please type into the chat box where you're joining from today so that we can get a sense of who's on the line. I see we have Neil McKenzie, who's a PhD student at Newcastle in the UK, working on DNA data storage technologies. Somebody from SynBioBeta in Frankfurt, Christopher Schorsch, and Hui Ping, who's from Australia, but currently based in Germany. So a lot of great comments and in the chat. So if you have comments that you want to make or you want to speak to the panelists, then please raise your hand and we would love to loop you in. So I want to go back to this theoretical framework that we're building to allow the interoperability for researchers over distributed time, distributed locations, because everybody, actually, let's just go around and just see where everybody is located. Mark, where are you located?
I'm in Cambridge, Massachusetts.
And Daniel?
I'm in Dallas, Fort Worth.
And Peter? We can see you're here in the Bay Area.
Yep. I'm over in Menlo Park. Strateos has offices in Menlo Park, down in San Diego, as well as in San Francisco proper. And but we allow everyone to access from everywhere.
Great. And Diveena, you're in Boston, is that right?
Correct, yes.
And Steve Duke is in North Carolina, I believe, is that right?
It is, and we have team members at Rutgers, Montana State, and Florida Atlantic. So we're, as a team, even spread out.
And Susan, whereabouts are you?
I work with DARPA proper, which is headquartered in Arlington, Virginia.
Okay, great. So Susan, if you can help to describe because then this is just 6 people who are part of the team, but there's actually 23 organizations as part of this distributed team. So can you help us understand how geography plays into this, and then how this system that you've created has been, give us a sense of how geography has been solved by some of the things that you've been creating through the SD2 program.
Right, as you said, we have 23 performers teams, and some of those teams are actually divided geographically as well. It's a highly collaborative program, and we understand that science in general is moving into this collaborative nature. One of the ways we've actually worked around some of this distribution is through coming together on a quarterly basis, which is not possible for all scientists, but for the development of these technologies, we've really had to rely on some of the traditional social structures that we build as scientists in order to build the technology that will eventually help mitigate the need for coming together and talking in the same place. And so SD2, as I alluded to, is divided, has taken the portions of science, the design, the experimentation, the planning, the analysis, and divided that up into different organizations in a way that forces us to take those communications and find ways to make them easier throughout this distributed locations.
And Peter, if you could just talk about Strateos for a second, because the whole point of Strateos is that it's this software-driven tech company that's serving the life science industry by providing the dendrites between different parts of the organization so that data can flow in the right direction, knowledge can be driven by data. Can you help us understand the role that Strateos is playing here, particularly around the geographic distribution of everybody?
Yeah, of course. One of the big things is, Strateos has been able to integrate ourselves really well inside of the SD2 network so that we can provide high quality lab automation, data and services for the scientists across that network. So no matter where you're located, and no matter what organization you're a part of, we've been able to use the SD2 system to allow scientists to submit experiments and get data back. I think one of the important things here, just to extend off of what Susan was saying, was when you're starting to think about moving scientific investigation into these large types of collaborations, one of the big things is we have to figure out how we talk about science and how we are able to sort of transmit that information from group to group and make sure that we're doing it flawlessly, basically. And so, we've participated both in using our interface, which is an open standard autoprotocol.org, as well as working with other groups to, make translations and integrate within the wider system as a whole. That means that we can get experiments submitted computationally as well as by scientists. It means that experiments can be submitted now through really easy interfaces. So I think we'll talk about this in a little bit, but Dan Bryce and others have been, Mark Weston and others have been making like Google Doc-based interfaces. So you can write your experiment in Google Doc and get it sent to really complex automation. And it also means when we upload data and get everything combined, and as Mark Weston was talking about in terms of the AI-ready formats, that those are all checked in terms of metadata, in terms of making sure that the science that we've done is accomplishing what the scientists like Steve and others have specified on the system overall.
Great. And I'm going to come on to some of the practical applications shortly, but Steve and Diveena, as two people closer to the biology, can we talk a little bit about the bottlenecks that you have experienced, and then maybe how this SD2 project has relieved some of the bottlenecks? So where was the pain, or where is the pain that you think a lot of people are experiencing in what they're doing? Steve, maybe you want to go first.
Yeah, let's start with that. So one of the things we're working very hard on is finding a mechanism by which the kinds of protocols that we use in a benchtop laboratory can be shared with Diveena or with Peter and onboarded in a more automated or semi-automated fashion. And so I think it's going to be really important for those of us who are doing bench science to be able to make that handoff seamlessly, and so that we can all produce data that in the end, nobody should know where it came from. So data coming out of my lab should look like data coming out of Strateos for all of our analysts. And so this is something that's been challenging, and we're making some headway on in the program.
Great. Diveena, any thoughts?
Yeah, so some of our biggest challenges, like Steve mentioned, is really also agreeing on kind of what are good quality data versus bad quality data. Different performers with different backgrounds and different applications have different standards for what they would consider good or bad. So really standardizing on those, essentially speaking the same language, or have a really good framework of understanding. And the other really kind of big hurdle that I think this program's done really well on is metadata, providing the context for any given experiment. What are the important variables in an experiment that you need to capture so that somebody downstream of the person doing the experiment can actually use the data in a meaningful way. And it's not just a lab person that needs to determine what those values are, but it's really a collaboration between the labs and the experimentalists to define, what it is that you should care about, what it is that you should capture and standardize. So I think this program's done really well in addressing some of those over the years.
And Diveena, I know quite a lot about the history of the founding of Ginkgo. So you live and breathe this stuff. So it must have been a pleasure to join this project. Tell us a little bit about kind of, you've been thinking about this from a long period of time. So are you educating other people on the project about best practices, or are you all collaborating and coming up with better ways to do things?
Yeah, no, I think the program as a whole, Ginkgo has been able to add values to the program through helping folks in the program understand how Ginkgo approaches an experiment, does an experiment, help educate. My background is next generation sequencing data analysis. So when they do DNA and RNA sequencing work, as a computational biologist, I can tell them, hey, these are QC values you need to worry about. These are data types that have these pitfalls, or these values that are important or not. And I think the reverse, which is everything we've experienced through SD2, when I'm working with my colleagues at Ginkgo, I'm able to also translate, hey, we've done these efforts within SD2, and we know this has worked and this hasn't. And so we also need internally has helped us improve some of the ways we think about data and standardizing. So it's been helpful both for us to bring what we know, as well as help Ginkgo understand how others are using what we've developed.
Great. And Mark Weston from Netrius, if you could talk a little bit about Netrius. Have you, when you're coming into this project, I presume that Netrius doesn't just work with biological data. Have you got other projects where you use other kinds of data?
Yeah, that's interesting. Prior to working on STREAM2, we actually had a lot of experience and background in managing cyber data sets, right? Which is fundamentally very different, right? There, you're looking at typically well-formatted, enumerated data sets. It's cyclical. You have nice, clean sequences. Biological data is you get incredible amounts of variation, right? And so one of the first things that we did on the program was start to model in the form of an experiment request what we would expect the experiment to produce. And this includes all of the conditions and all of the data that came out. And we started to compare that to what the different labs are producing. We did this in a lab agnostic fashion. And we started to see some very interesting things, whether it's, could be a reporting error, could be something changed on the bench, right? And so I think a key lesson that we had to apply there was that our system had to be flexible. It had to be able to change and to adapt and to account for things that would come up either during execution or actual, quote unquote, errors or, discrepancies they would see in the data. At least we'd be able to explain them. In the absence of this, we would just get experiments run and we would get reports out, but we didn't really have a good sense of whether what we received is what we expected.
And when you were coming into this project and you're looking now at the kinds of biological data, how does it compare to the kinds of data that you're getting from cybersecurity? And then, what was your first impression? And then how has that kind of changed over time? And I see Steve smiling, so I'm interested in
Yeah. I mean, heterogeneous is a term that people toss around, right? And I think fundamentally, if we just look at the different labs, even the labs we have here, everybody's going to call things a little bit differently. They might have an internal term or a lens, or they might use this ontology or that ontology, right? And so it's very challenging just to get even a common ground to say, okay, like this is the strain that we're working on. This is the experiment, and everybody can know kind of that we're talking about the same kind of thing. And so without requiring these other organizations to change what they're doing internally, we developed translation software that basically takes all the different metadata formats from all the different labs, runs them through and harmonizes them into a single format that then is piped downstream to all the analysts. So the analysts benefit because we solve a whole bunch of problems upstream that they don't have to deal with and wrangle kind of post hoc once they get the data. They have nice, clean data frames with terms that are normalized already to go.
And it's so it's kind of sounds like 90% is prep and then 10% is being able to run it. And Daniel, I'll come on to you shortly about talking about symbolic logic and AI, because that's where the beauty comes in of being able to now the rubber hits the road. But Mark, can you explain how does the Strateos platform tie into to what you're doing?
Yeah, Strateos is excellent. I mean, we have a fully automated loop now where we can go from not just the experiment request that gets submitted, that'll run at Strateos, that gets uploaded. Further automation then kicks in, processes all of that data, and then we run several first stream analyses on those data sets as well. So we've incrementally, as the program has progressed, automated, piece by piece, entire cycles of the experiment design, build, test loop as it is. And we don't have the system fully running by itself. It's not a self-aware AI system that's designing the next piece, but we're automating out a lot of those, little basic bits. And we're letting the humans, bring in their expertise, bring in their subject matter expertise to figure out, okay, diagnose what's going on here, design the next phase of the experiment, get that loop turning faster and faster so that we can do better science. And Strateos' ability to run these automated experiments at scale has been critical to that.
Great. And Peter, you mentioned a website earlier. I think it was protocols.io. Could you just type that into the chat box for anybody who's interested, and also the Strateos link for anybody who wants to follow up on Strateos?
That's great. Will do.
And Daniel, so now comes to the fun part where you actually get to apply the AI. Now you've got all the data. The experiment that you're doing, you've got it all in the right format, everything can talk to each other. Is this where you come in terms of symbolic logic and being able to run the AI?
Yeah, and I appreciate you saying it's the fun part, because I think it's the fun part too. But there's really kind of two camps of AI that really come to bear on SD2. There's more of the traditional symbolic AI, which is really about doing logical inference and automated planning And that comes in kind of on the front end where we're doing the resource allocation, taking a sort of the what of the experiment and figuring out the how to accomplish it, and translating the sort of abstract description of the experiment into something that can actually be run automatically at Strateos. So that's really a place where we brought a lot of expertise with symbolic AI, and we have a lot of other defense customers, and this has been applied to other things like military logistics, autonomous vehicles, and it's really some of the same machinery that can be applied here. Then on the other side, once you've produced data, that's really where the machine learning comes in, and we can do a lot of things that we couldn't do before. So one of the things that really I found that machine learning was helpful in SD2 was we were able to, in some cases, remove some of the preparation steps in protocols by using machine learning to supplement some of the same information that we'd be getting out without having to do these preparation steps. And that actually has allowed us to reduce the cost of experiments by just using machine learning techniques. And I found that to be a really exciting part of the program.
Great, thank you, Daniel. So we have a few questions from panelists, which I'm going to come to shortly after we talk about the applications. And I'm going to invite you up to put your video on and speak to the panelists yourself. So I'll invite Ben Keller and Ravi Ramanathan. Sorry, Ravi, I know we know each other, but your last name always gets me. And Davey as well, who's at Cal. So we'll invite you all up, and you can ask your questions live. Some really good questions there, or make a comment. Ben, I think you want to talk about the University of Washington Biofab, which will be great to learn about. But before we do that, we've talked again about this wonderful theoretical framework. So now let's talk about some of the projects, some of the applications, some of the pathways that you've been designing and optimizing. And Susan, maybe you can help us understand. My question when we were doing a prep for this event was, how did you choose? And so you kind of had a democratic way to choose which things that this whole piece of machinery that you built was fired at. So can you give us a little bit of background to that, and what were some of the application areas that you were interested in?
Right, yeah. So when the performers came on, there was a process led by our integration team and integration and evaluation team in order to basically see where people's natural interests lie, and then to coalesce around shared interests in a way that we can project those forward. Again, everything is dependent on this collaborative nature, so sort of forcing everyone into given sets did not make sense. And so what we ended up coalescing on within the synthetic biology group was one group that was focused on yeast biology and creating circuits in yeast, and the other was on bacteria and specifically exploring how to move circuits and other designs into novel chassis bacteria. And so being able to easily take the tools that we've developed in one chassis and be able to quickly move to a different organism.
Great, excellent. So Steve, can you maybe delve a little bit deeper into your favorite project and help us understand, what it was about?
Yeah, sure. So I've been part of the project involving engineering circuits in yeast, and I've been a yeast biologist studying yeast cell division and growth for 25 years, and so that was a kind of a natural place for me to land. And I think that the approach, and basically you've heard it, was to onboard a design, build, and test loop in which we would design purpose-built circuits. For the most part, they were set up to sense certain molecules in the environment and then respond. And the measurement of the response was GFP, and using flow cytometry or plate reader data to decide whether or not the circuit did what it was supposed to do. One of the interesting things that we found early on is that it was really hard to get reproducibility with the circuits we were looking at right away. And one thought was that small changes in growth conditions might actually affect how those circuits operate, because it likely affects the parameter regimes in which these circuits are operating. And so we actually did an exploration of growth condition space in this program, and spent time asking, to what degree were the circuits robust to these different growth conditions? And could we actually design circuits that would be robust to various growth conditions? And so we had design tools, we had strain construction tools, we had analysis tools, and then diagnosis tools to try and figure out if a strain didn't do what it was supposed to do, why not? And so this entire loop got put together in the program to design and test purpose-built circuits in budding yeast.
Fantastic. Excellent. Well, good to see some practical use. And Steve, you described yourself as a systems biologist, and now you're working with synthetic biologists. So you're in the perfect position to now take natural circuits and now take engineered circuits, run them both through this amazing analysis platform, and then see what the results are.
Yeah, that's exactly right. And we had actually had a tool called DSGRN that was able to computationally analyze the robustness of a particular circuit topology in across parameter space. And once we realized that we had a tool to analyze that, we could turn it into a design tool that we could actually test designs computationally before we went through the pain of actually building the constructs and running them through this entire loop. So so that's proved to be a rich area of research.
And this is the holy grail of bioengineering. This is what we're all looking for, is being able to model something before you have to do the experiment. So
and that's the tricky part with biology. And engineering usually have a better idea of what your parameters are when you build a circuit. And in biology, we had very little idea of what our actual parameters are. And so that's one of the kind of dark corners of synthetic biology, I think.
excellent. And what was one of the most surprising things that you found from the results?
Wow. Yeah, I think the surprising thing for me was that it was hard to predict a particular growth condition in which things would fail. Some things didn't do very well in what we normally think of at the benchtop as the rich media condition, and they did better in some conditions that were a little more harsh. And so one of the things that we were aiming to do is try and predict which regime these circuits would excel in and which they wouldn't.
Very interesting. And Diveena, what about you? What was the most surprising thing that you uncovered?
Not entirely surprising, but also definitely reiterating what we've already known. Biology is full of exceptions. That we would make all these designs and on paper and in theory, and some tests in our in partner labs. When they do it on benchtop, it works. And then when we try to do it in high throughput, some of them fail, and trying to understand which variables specifically contributed to failure. That's hard. Yeah, so not entirely a surprise, but definitely it's been like being able to learn and reiterate. And this is where, like, Ginkgo's high throughput pipelines are so useful, because we can synthesize, try to build again and repeat. So we're not entirely paralyzed if things do go wrong. But yeah.
Great, excellent. Ben, do you want to unmute yourself, and if you want to turn on your video, you can, and maybe just introduce the bio hub, sorry, the biofab at UW.
Yeah, so obviously I'm not at the University of Washington at the moment, but we are, our group actually, I work for Eric Clevens, who's a professor and now the department chair of electrical engineering at University of Washington. And Eric developed a system a while ago where it's called Aquarium, where his goal was to automate the work that was done in his lab. And because his grad students would do experiments and they would, they'd do a strain construction and it would fail, even though they were following the same procedure as somebody else. So the question was like, what does same mean? And so what Eric developed is this system aquarium that has protocols that are well-defined and actually written as code and then executed, very much like what you see at Ginkgo and Strateos, right? So, except the emphasis is actually on doing bench work, where, you know, Strateos, you can see, you go in and you see the work cell, you know, moving the robot around and all this stuff. We have actually in the, you know, BioFab as a service within the university actually does the work. You know, you see them running pipettes based on the screen. So that's the base story of where we came from and where we fit. Sorry.
And yeah, and Ben, you're part of SD2. So what you're describing is that, yes, you know, the whole point of a platform, let's say an integration platform like Strateos, or this whole abstraction hierarchy that we're creating, is that different parts can slot in. There can be an automation part like Strateos, but then you can still have the I was going to say the painful manual bench part. I know that the system that you've developed is slightly less painful, but I've heard it's more like a McDonald's ordering system,
right,
for doing lab bench work. But yeah, so sorry to steal your thunder, but that's the basic principle, that different pieces of this can slot in. Is that right?
Right, exactly. And what we've done, what we realized is, I mean, originally we were just being like Ginkgo and Strateos in terms of like doing work, and we do a lot of protein design, you know, runs and assays. But mostly what we're doing now is building our software so that people like Steve can use it in his lab, and then also building the integration, you know, collaborating with Peter and Dan and Mark in order to build the integrations so that Steve can look like Strateos to SD2.
Great. Excellent. Well, thank you for contributing, Ben. Appreciate it. And you're welcome to stay on and join the discussion. I want to bring in Ravi. Ravi, did you have a question or comment? And maybe go ahead and introduce yourself, because you're dialing in from India. I think it's late night there, so appreciate you joining us.
Yeah. Hi, John. Thanks for the excellent webinar. Yes, myself, I'm Ravi. I'm from Chennai, South India. I've been working in Synbio and AI, doing more on the protein design side, protein folding, dynamics, as well as design. We're building a stack for it. Of course, we're not having sophisticated automation tools here, which is something I would like to also build here in India. So my question is, are these software stacks being open source, or some may not be, but in general, Ben mentioned that aquarium is, but I would like to know how much adaptable it is for some labs that may not have all the automation platforms roped in, but can still benefit from the analysis part and all that.
Perfect. And for the panelists, if you just want to unmute, and then I'll know that you want to take that. Yeah, Susan, go ahead and answer.
Yeah, so the SD2 program is a 4-year program, and we're 3.5 years in, which is to say that most of the components being developed on the SD2 program are available open source in some capacity, although some of them are still in development and are not fully available yet. But interest in using the tools, by all means, reaching out to us. You know, I think I can speak for everyone here. It's our goal at this point is to get what we've developed in SD2 sort of out into the public and show how these tools can really help speed the efficiency of what we do in synthetic biology.
Great, thanks, Susan, and thanks, Ravi. Vicky, I believe that you're dialing in from Australia. Am I correct? Vicky, you're on mute. If you're able to unmute, we can ask your question. Otherwise, we can come back to Vicky in a second. Davey, you have a question about the future. Go ahead and unmute, and if you want to turn on your video as well. Great, good to see you.
Yeah. Hey everyone, I'm Davey. I'm near the Berkeley campus right now, so not too far from a couple of you. And my question also comes, I guess, from the perspective of being a student and from the perspective of like, what skills should I learn? And that is, where do you think the areas of greatest change will be at the intersection of AI and bio? And are there any, I don't know, do you have any predictions for where we will be in 5 years?
Who would like to take that? Mark, do you want to take a stab? What are your predictions?
Yeah, I'm going to approach it from a slightly different perspective. I mean, we've even within the life cycle of this program, we've been able to actually automate some experiments, right? So the fastest experiment is one you don't have to run. So there's some characterization work that we've been able to do. For example, looking at a new host that we're trying to transfer something to, and you could run all those experiments, or we could take all the previous hosts that we've run before, or look at partial circuits, right, and combine those up into dual inducer circuits, right? So we're able to actually predict the outputs with increasing accuracy as more and more data builds up, right? So I think as we see more of a collaboration, more of a data ecosystem build up in the synthetic biology community, you'll be able to, with increasing confidence, predict these experiments. And rather than just kind of running, characterization type things, you can go on to tackle harder and harder problems.
Would anybody else like to take a stab? Maybe Diveena, what are your predictions? And then Steve, and then Daniel, and then Peter.
I'll answer it from a slightly different flavor in terms of, I think synthetic biology as a field, although in a large way is being applied to life sciences, I definitely see a future for synthetic biology in areas that we don't think about very much, areas like material sciences or climate change mitigation efforts, not just engineering biological system, but being able to apply it to big problems. So I think no matter what field you are in or whatever field that you're interested in being in, I think that there likely is a way to apply synthetic biology to it. And in terms of your question on what skills you should be learning or developing, I think a lot of folks are focused on computational skills. A lot of people have programming backgrounds, and having a kind of a background not entirely focused on one area, but having some biology, having some computer science, having some other relevant engineering type background. It could all be very, very helpful moving forward.
Great. Daniel, what are your thoughts?
Yeah, so one of the major challenges we've had is really in representation. Knowledge representation is a big part of AI, and it's been kind of glaring when we approach ST2 that you need to capture what it is you did and what you did it to, and in order to apply all these AI machine learning techniques, you really need a good capture of what happened. And it's really difficult to capture that in a kind of clean way. And so I see knowledge representation as being a big thing going forward for SynBio.
Great. Steve, what are your thoughts?
Yeah, I think the idea of as somebody who has a long experience in doing bench science, the distributed science is where we're headed. And I think the reality is to produce high quality AI ready data, we can't take data from labs producing it in slightly different ways with slightly different measurements, and all of these things need to be standardized. And so I expect that going forward, we're going to see a lot more interaction between benchtop laboratories and laboratories like Strateos or the BioFab or Ginkgo. And in terms of skill sets, I'll echo what Diveena said and add one thing to it, and that is learning how to work in an interdisciplinary team is a really important, underestimated skill, because what has made SD2 successful is the ability of folks from the front end on experiments through data and metadata all the way to analysis, and then back again to the experimentalist. That integration is key for success. And so working in teams, learning how to work with folks outside of your area of expertise, I think is going to be one of the most important skill sets going forward in the next 10 years.
Great answer. Peter, what do you think?
Yeah, I love questions about the future because it's very hard to be wrong. I think we're all excited to answer those types of things, so thank you for that. I think about three general directions. One, as has been mentioned, tighter integration between different labs, being able to seamlessly communicate and basically be like, well, data coming out of all these different labs are all basically the same, right? And so, we've been working within this group and all of these panelists on how do we make that better. The second part is that closed loop science. So how can we integrate more automation, more AI-enabled processes towards accelerating science so that we can start making decisions in a very tight loop, based off of like data generated, that updates the model, that creates the next experiment, that automatically executes that experiment, that generates more data, etc. And then third is, now that we have all of these resources, really starting to think about doing science in different ways. And Mark's mentioned some of this already in terms of being more able to do more in silico predictions when you're testing hypotheses, but also thinking about exploring science in more parallel manners. So right now that you have access to high-throughput screening tools, the ability to screen 1536 wells, the ability to screen tens of millions of micro droplets at a time, et cetera. Really thinking about setting up experiments so that they can be parallelized and processive. I'll add on just to the list of things that you should learn is just learning to learn, of course. It's I think it's a huge enabler because this intersection and the interfaces of all of these fields are moving very quickly. And, we're starting to think about these larger concepts. Can we take synthetic biology? Can we, move it towards the point of care so that, we can start producing things, out towards the edges? Can we distribute that production better and things like that? But learning to learn really sets you up to be able to be flexible and take advantage of the changing interfaces and intersections.
That's great. Thank you, Peter. And if somebody is listening or tuning in and they're wanting to take the first steps in trying to set up a similar system like this, and Strateos, of course, enables that. This is the service that you provide. And of course, Strateos is the sponsor of today's event. How do they get in touch? What should they do? What should be the first step that they take?
Yeah, I welcome people to visit the Strateos website and feel free to get in touch with me. I'm peter.lee@strateos.com. And I'm happy to also connect people to the rest of the SD2 interfaces as well and talk about all of the work that the entire system has been building. So we are both a service lab, so we can take on experiments, and we can also deploy our software for automation and get labs running like ours.
Great. And I also wrote a really nice piece about Strateos and starting a startup from your coffee shop. So if we've got any budding entrepreneurs listening today, maybe we can post that in the chat, that article. It's quite a nice feature of how you can start a startup, get to Series A funding, get the data you need, but not have to employ a scientist or run a lab or buy expensive equipment. So I encourage you all to try that. And also we have the GitLab repository, Mark posted into the chat. So I want to make sure that people saw that. And we've tweeted out.
GitHub, sorry, GitHub, not GitLab.
Oh, sorry, GitHub repository, thank you. And we also tweeted out, which I think is the quote of the webinar, that the best experiment is one you don't have to do. I love it, Mark, so thank you. Susan, did you want to not be wrong and talk about the next 5 years of bio and AI?
Yeah, I just wanted to add a few extra thoughts here, which is to say, I think sort of the next advantages in bio and AI really come down to the data we're producing. And if we think about data standards, it's really the interoperability and the reusability that are still challenging. And to think about where that can take us to be able to use data in areas that appear to be unrelated right now. And in order to be able to do that, we need to be encoding the metadata, we need to be encoding the process and the information into that data. But we also need to be making sure that the data is generally usable in a way for things that we can't even predict today. And so that really comes down to, again, the data, the metadata, and the protocols that we're encoding as part of that.
Great. We're getting a flurry of last minute questions coming in, and we'll try to take as many of them as we can. Vicky Forrest, it sounded like you were on the move there, but if you want to unmute and ask your question, please go ahead. Otherwise, we can come back to, yeah.
Hey, John, thanks. I'm really amongst enormous roadworks now, even worse than when you asked earlier. I'll come back later if I can.
Yeah, sure. Just unmute and I'll know that you're ready to come back.
Thank you. Thank you.
Yes. Okay. So we can come back to Vicky. Huipeng Lim, would you like to ask your question? You could unmute and turn on your video. Otherwise, we can ask Neil Mackenzie or Varshith if you just want to unmute, and I'll loop you in.
Hello.
Hey Neil, how are you?
Yeah, I'm not too bad. Just in a grey Newcastle. What I was really curious about.
Yeah,
was,
Neil, go ahead and introduce yourself to the panelists and then ask your question.
So my name is Neil Mackenzie. I'm a PhD student at Newcastle looking at DNA data storage and associated technologies. And what I'm kind of really curious about is the beginning of all of this. So going from a paper protocol to an automation system is how many people are looking at how you convert a paper protocol to a machine protocol to then put it through so that when you get your results, you can then automatically change those variables and therefore get a full closed loop system.
Who wants to take that on? Maybe Steve, do you want to have a stab at that?
Yeah, this is a question I've asked pretty much daily since the start of the program, because as a bench scientist, I have a stack of protocols sitting in my office. Some of them are on the back of cocktail napkins. And the real question is, how do we take those and onboard them in a way that I could use them in my lab, send them to Peter at Strateos, or Ben at the BioFab, or Diveena at Ginkgo, and have them run the experiment and get the same kind of data and the reproducibility out of it that one would hope. And so there are efforts going on that I think others can speak to here.
Yeah. One of the things that we've been working on within the program is an initiative called the Common Protocol Representation. So this is really thinking about how each of the different performers from Steve's lab to Ben over with Aquarium and BioFab to Diveena Ginkgo to us at Strateos are expressing protocols, and can we get to a common platform so that we're all expressing things similarly and have bridges to each other. Part of that initiative is to start looking at, can we parse documents and convert them into this common protocol representation, and also the reverse. So, can we take common protocol representation and create documents easily and get them posted and hosted in different places? I will point out, one of the things, and I mentioned in the chat, is I think it's pretty amazing that we have Google Docs that you can write that initiate science, right? So that there are basically there's a whole workflow. It describes what the workflow is. There are blanks and parts of the chart and forms that you can fill out, and that controls things on the back end so that automation and benchtop work and all of this is happening to surface the data and get it back into the ecosystem. So there's a lot of cool stuff there. If there are people in attendance that are interested in that, we're starting to develop these things out. We'd love to get more contributors in the future as well. So please reach out and we'll coordinate that.
Is that sort of work robot agnostic? So i.e., if you're using an open source platform, for example, it's much easier. So say, for example, an OpenTrans, you can just Python all the way. Whereas if you're using an Analytica Genica, they use their weird and wonderful file format for their protocols. Is there any aim to try and be able to have a robot agnostic overall pipeline platform?
Yeah, that's a great question. Really insightful. Yeah, that's definitely the aim here is for the common protocol representation to really represent scientific intent. So it's coming from the scientific side. And it's kind of, like what Steve is asking for, right? Like I talk in science. I don't want to talk in your robots. I don't want to talk in, these specific domains. So really thinking about, right, how do we drive these actions from a robot agnostic, from a laboratory agnostic, right? From a, work cell or consortium of robots type viewpoint. So really thinking, and some of the roots of this you can see in autoprotocol.org. These are some of the basic actions that we're looking at integrating into this common protocol representation and then creating larger workflow languages around that. But it's based off of looking at scientific intent and breaking it down so that you are basically looking at like physical principles and not, a device does this. So like a centrifuge, rotates at specific RPM or something like that. It's like the motor moves or at a specific, RCF basically, and not like a specific rotor configuration, things like that.
Excellent. Thank you, Peter, and thank you, Neil. Hui Pang Lim, would you like to go ahead and ask your question? Introduce yourself and ask your question. No, we're still missing Hui Pang, but Varshit, would you like to go ahead? Oh, there we go, Hui Pang. Can you, we can't hear you. Can you try speaking? Yeah, we can't hear you for some reason. It says you're not muted, so but let's take Varshit's question and then we can come back to you, Hui Pang. Go ahead, Varshit. Introduce yourself and ask your question.
Sure, thanks, John. So I'm Varshit. So I work in AI and data science engineering at an e-commerce company called Wayfair. But I was involved with computational synthetic biology back when I was doing my master's, part of Douglas Densmore's group.
Just speak up a little bit, Varshit. It's a little bit difficult to hear you.
Oh, sorry about that. Am I audible right now?
Yes, much better.
Perfect, perfect. Yes, I work in AI and data science engineering at Wayfair currently, but I was involved with computational synthetic biology a few years ago while I was doing my master's at BU. So my question was, so at that point in time, like I was kind of introduced to SBOL, which was the SynBio open language standard. And I know that standardization was like brought up in the discussion as well a lot. So I want to know, like, where is the field heading in that direction, and then to ease out integration, and also like some of the research that has been done in universities, like how easy it is for me, like who is actually not part of a research group or a university right now, but still want to like work with, innovation that has been happening in the field. So what should I do, like, to keep up with what is happening in terms of standardization? And if I want to like build something or contribute something open source, how should I be doing that?
Great. Susan, would you like to take that one?
Sure, and then I might bounce this to Dan as well, which is to say, yes, there's work been involved in continuing the development of the SBOL formats and the idea of standardization. Dan, you want to pull off?
Sure. So with SBOL, we've been using that as one of the main languages within SD2. And with SBOL 2, it started out really representing the constructs that we'll be experimenting with. And we've moved on to SBOL 3, which is more concerned with capturing the experiments involving those constructs. And so as much as possible, we've been trying to use SBOL as sort of the driving force for representing, I mentioned before about knowledge representation being important for representing experiments. And this is really the language that we've been using to do that. And so it's really been building layers on top of that language to capture more and more of the needs in the program. And so we see that as being something we'll continue to be involved with in the future. The other types of standards we're looking at are, we mentioned the Common Protocol Representation Language. We're working on that as a potential standard as well to facilitate lab interchange. And those are really the main knowledge representations that we've been pushing as sort of technologies out of SD2. And in terms of being involved, I think both of those are open standards and there are ways to be involved in those. But if you had more specific questions, I could certainly answer those offline if you want to reach out.
Great.
Absolutely.
Thanks, Dan. Thanks, Varshit. If Dan, if you want to post your email into the chat as well, if you want Varshit to reach out to you. Vicky, go ahead and ask your question.
Hey, thanks very much, John. More of a comment than a question. I'm Vicky Forrest. I'm the Commissioner for Trade and Investment Queensland in the state of Australia, but I'm based here in San Francisco. And I was out on the noisy streets of San Francisco when you caught me earlier. But really, the primary reason for my involvement today is to understand the types of tools and support that Strateos provides for young SynBio companies so that I can be a source of valuable information back to the many early stage SynBio companies in Australia and in Queensland specifically. And the second thing that I'll note here, and John, this can be of use in other sessions for you, is Queensland's activity in establishing very large capacity precision fermentation for these SynBio companies as they grow and scale to require that. And Queensland's ideally located for many reasons: feedstock, proximity to Asia, and many other things that I'm happy to talk to companies about as they get to that stage of their development.
Wonderful. Thank you for joining us, Vicky. I'm sorry, Hui Ping and Harley, we have run out of time today, but thank you everybody for joining us. Thank you to all of our panelists, to Dan Bryce, to Peter Lee, to Mark Weston, Diveena Becker, Susan Buckhat-White, and Steve Hasse. And thank you to all the great comments, and particularly thanks to Strateos for sponsoring today's event. And Strateos, as you know, is a software-driven tech company, and it's serving the life sciences industry. And as you can see from all the examples that we've given you about connecting to data, being data-driven, connecting computation and automation and high-throughput robotics, it is a software platform that can connect it all out. If you want to get in touch with Peter, his email is in the chat. You can go to strateos.com, and you can take all of your biology and all of your robots into the cloud with you for the next 100 years, the century of biology, which is what we're welcoming in. So thanks again to all of our panelists. This has been a fun first event of the year, and sign up to the SynBioBeta newsletter if you're not already subscribed to find out the next event we do. We have a big Food and Ag event next Wednesday, and we have a farmer event in May, and then we'll be back in person at the Oakland Marriott the first week of October. Everybody's looking forward to getting their vaccinations, bringing their vaccine cards, and partying like it's 2021. Thank you all for joining us, and we will see you all again soon. Bye-bye now.